dear student, how are you? We people have discussed application of polymerase chain reaction in last video, right? I hope you people understood that really very well. ठीक है, आपने अच्छे से उसको देखा है, उसको पढ़ा है. In today's video, we are going to start with your bioreactor. Okay, dear student, if I am talking about your bioreactor, so these bioreactors are also called as fermenters. Over here, I am writing it as a fermenters. ठीक है, and these bioreactors are applied. These bioreactors are applied, or you can say used to obtain our foreign gene product. Am I clear? अब ये सारी प्रोसेस हमने क्यों करी है? What is the reason for the same? To obtain product, yes or no? Yes. And generally we are talking about proteins. We are talking about proteins. When you are going to insert gene, right? That gene undergo replication. Yes, it undergo transcription. It undergo translation to form proteins, and our desired product is recombinant protein, which expressed in some heterologous host. ठीक है? For obtaining the same, there is a large vessel. There is a large vessel which is called as your bioreactor or a fermenter. So bioreactor or fermenter basically है क्या? You can compare it with a kind of dabba only, right? In which you are going to put snacks in your home, right? So once again, your bioreactors are large vessel in which raw materials are biologically converted into products, by microorganisms, animal cells, plant cells, or even enzymes obtained from the same. ठीक है, नहीं समझ में आया? मैंने ये बोला आपको, बायोरिएक्टर है एक वेसल जिसके अंदर हम रॉ मटेरियल डाले, एंड दैट रॉ मटेरियल जनरली बाय मींस ऑफ माइक्रो ऑर्गेनिजम्स आर कन्वर्टेड इनटू प्रोडक्ट्स. ओके, इट कैन बी माइक्रो ऑर्गेनिज्म, इट कैन बी एनी सेल, मतलब प्लांट सेल और एनिमल सेल, इवन इट कैन बी एंजाइम. जनरल आई एम टॉकिंग अबाउट. एम आई क्लियर? सो in your bioreactor, we are processing raw materials for obtaining products. Am I clear? If I am talking about this culture, what is culture? हमने recombinant DNA तैयार किया, वो recombinant DNA को आपने डाला E. coli के अंदर, right? Now we want to allow this E. coli to multiply so as to our gene of interest also multiplies, or product we will get. Yes or no? Yes. तो ये माइक्रो ऑर्गेनिज्म को हम डाले कल्चर में. Now what do you understand by the term culture? Culture is nothing but medium, nutrient medium, you can say. Where your microorganisms uses this nutrient for their growth, I mean to say for their multiplication, for their reproduction. Am I clear? So if we are using small cultures, then definitely we will not get much product, right? So for obtaining large scale production, हमको चाहिए ज्यादा culture, and this is what we are obtaining in bioreactors. ठीक है? Now if I am talking about your bioreactors, types of bioreactors, then the type of bioreactor which is commonly used is called as your stirring type bioreactor. What it is said? It is said as your stirring type bioreactor. Okay, this stirring type bioreactor are basically classified into two categories, as we know that. Number one is your simple stirred type. Okay, over here I am writing is your simple stirred type, right? आपको अच्छे से पता है. And second one, if I want to say, is your, yes, parched stirred type. Simple as that. So second one is your parched stirred type. Am I clear? Okay. Dear student, you know what is the difference between simple stirred and sparge stirred. If I am saying your sparge stirred bioreactor, तो इसके अंदर sterile air bubbles are sparged. Am I clear? What is sparge? We very well know that it is your plate which have pores, through which if we blow air there will be formation of air bubbles. क्या बनते हैं? Air bubbles बनते हैं. And this air bubbles, ये जो air bubbles यहाँ पे, so what I am writing in this case, air bubbles are sparged. Am I clear? मतलब air is blown through a plate containing pores, and due to this reason there is formation of air bubbles, and this air bubbles increases सरफेस एरिया. ये क्या इंक्रीज करते हैं? सरफेस एरिया इंक्रीज करते हैं. Am I clear? So in this video I am not discussing diagrams of your simple stirred and sparge stirred. Please visit the video that we have done in our regular classes. ठीक है? चलो. Now here I am going to focus on a process called as your fermentation. Process called as your fermentation. Okay. So if we are talking about fermentation process, so what do you exactly understand by a process of fermentation? By this process, जो raw materials है, वो microorganism use करते हैं. Raw material in the sense here I am talking about mainly glucose, and they convert the same into alcohol. This is the process your fermentation means. नहीं समझ में आया? Once again, what is fermentation? The process in which microorganism converts raw material, that is sugar, that is glucose, into alcohol. ये process को कहा जाता है fermentation. So what is fermentation? It's a process by which your microorganism converts raw material. To be more specific, I am saying glucose into your alcohol. ठीक है? This is what I mean by fermentation. यहाँ पे कौन होंगे आपके? Microorganisms. Over here there will be presence of microorganisms. So dear student, if I am talking about fermentation, right, then generally ये जो fermentation word है वो किसके लिए use होता है? For an anaerobic process. For an anaerobic process, but nowadays it is used for both aerobic as well as your anaerobic one. ठीक है? In this process, proper care, proper condition, I mean to say sterile condition should be make available for getting desired product. ठीक है? तो यहाँ पे कैसी condition maintain करनी है? Your sterile condition, you can say. Okay, so now if I am talking about your fermentation process, so at the end you will get product, and product can be cell itself or some of the product which is produced by that cell. ठीक है? So here if I want to say product जो मिलेगी वो क्या मिलेगी आपको? Cell itself, or it can be some product produced by cell. Am I clear? चलो. So it can be any cell product. It can be any cell product. ठीक है? Now if I am focusing on types of fermentation, so it is broadly divided into two categories. Number one is said as your batch fermentation. ठीक है? And second one is said as your continuous fermentation, as the name suggests. यहाँ पे कुछ batch form में होगा, यहाँ पे कुछ process continuous होगी. Now what is this? If I am talking about your batch fermentation, so in this case, in a bioreactor or a fermenter, what you are going to add? You are going to add raw material, okay, along with microorganism. My dear students, raw material in the sense I am talking about your culture, it's your nutrient medium. जैसे कि यहाँ पे मैंने लिखा है ग्लूकोज, क्या लिखा है यहाँ पे मैंने ग्लूकोज लिखा है. So in batch fermentation you have to add a raw material over here, right, along with raw material along with your microorganisms, as you can see. Yes or no? Yes. And you are going to close it. You are not going to open it from outside. आपने एक बार बंद कर दिया मतलब बंद. In no way you are going to open it. Okay. Once raw material is done, once raw material is over, it is opened and product is taken out. Simple as that. So this is what I mean by batch fermentation. नहीं समझ में आया? मैंने बोला एक डब्बे के अंदर आपको डालना है रॉ मटेरियल, आपको डालना है माइक्रो ऑर्गेनिज्म, फिर वो डब्बे को हिलाना नहीं है. You are not going to disturb that bioreactor. ठीक है? Once product is formed, you are going to open this and you are going to remove your desired product. Simple सी बात है. Okay? Whereas if I am talking about your continuous fermentation, then in this process your nutrients, or you can say it as your raw material, is being continuously added and products are being continuously removed. It is removed as fast as it is formed. It is added as fast as it is consumed. Are you getting my point? So there is a kind of circle. एक तरफ से डालो, दूसरी तरफ से निकालो. What you are going to add? You are going to add raw material. What you are going to remove? You are going to remove products. ठीक है? I hope ये आपको समझ में आया. Okay? Once again, continuous fermentation में क्या किया जाता है? Nutrients are being added as fast as they are consumed. Products are removed as fast as they are formed. Simple as that. Okay? So if you're talking about your bioreactor, then it is very easy setup, right? One can establish the same in laboratories. Okay? In this we are having different control systems, जैसे कि temperature control system, pH control system. We are having your foam control system we are having. Okay? And many more controls we are having. This all are the advantage. The only disadvantage of using this bioreactor is that they are too expensive. बस यही disadvantage है. They are too expensive to fit. Am I clear? So this is your bioreactor and this is the fermentation process which occurs in bioreactor. You can do it in two ways. Your batch fermentation and continuous fermentation. Okay? So I hope you understood this really very well. Thank you.